So, what is this thing called? A phospholipid. And this thing next to it is? Cholesterol. The phospholipid's made of two major parts, these blue things which are called glycerol, and two tails that are made out of saturated and unsaturated fatty acids. And they occur as a bilayer. Now this is an important part of our chemistry to remember. Do fats love water or hate water? Hate water. Fats hate water. Oil and water, they separate. Oil and water separate, so they hate water. Things that hate water are typically non-polar. Non-polar. Things that hate water are non-polar. So this part of the membrane, right there, that middle section composed of fatty acids, we call that non-polar or hydrophobic. Non-polar or hydrophobic. Hydrophobic. And the glycerol heads love water. They're polar, and we call them hydrophilic. So they are polar, hydrophilic. And these hate water. And they're hydrophobic. It's really important for that selectively permeable or semi-permeable thing that we talked about on Friday, where some stuff can fit through and others can't. The things that affect whether or not something is going to pass in or out of the membrane are going to be their polarity and their size. The spaces between the phospholipids here are pretty small. So very small molecules can pass through. Water can freely pass through the membrane. Not huge quantities of water, but molecules of water will pass freely through. Some small gases, oxygen, carbon dioxide, they pass freely through the membrane as well. Typically, it's non-polar things that are going to pass freely through the membrane because they have to go through this giant non-polar region. You guys occur late enough in the day, and quite frankly, you probably had to do it this morning as well. You had to walk through the seniors to get here, right? Beginning of the year, that was likely much more difficult than it was at this time of year. You guys are used to it now, right? Was it a comfortable passage for you the first time you had to walk amongst the seniors? And some of you are like, I have siblings who are seniors. It didn't bother me. Those of you who didn't have siblings or were unfamiliar with the seniors, true or false, walking through the seniors was scary. If you wouldn't use the word scary, how about intimidating? Made you slightly nervous. If I were to send you into a classroom right now that is all seniors and ask you to go ask another teacher a question, what would your comfort level in that room be? Not so much? It's the same idea here. Molecules prefer to be with other similar molecules. If I did the same thing to you for a classroom full of freshmen and said, hey, go ask Coach P a question, you'd probably be comfortable doing it, right? Like, yeah, whatever. It's no big deal. These are people I know, people like me. I know Coach P, whatever. Molecules behave similarly. They will associate with molecules who like similar things. So if you are a water-liking molecule or a water-loving molecule, you're a polar molecule, you would rather be with other polar molecules. If you're water-fearing or water-hating, you would rather be with other water-hating molecules. So that's why non-polar things can freely pass through this middle section, no issue. They're similar in chemical structure, so in and out, no big deal. Unfortunately, more than just very small or nonpolar things have to pass through the membrane. All sorts of stuff has to go through the membrane. Glucose, sucrose, charged things have to go through the membrane, so ions like sodium and potassium, they can't freely pass here. They won't freely pass there. So we have to have other ways to get them across, which is what some of the other stuff is in the membrane. Now you have this picture, right? Because we're going to start labeling and going through what some of these things are. So you have the picture? Some of you weren't here on Friday. That's why I want to make sure you... I don't know if you actually went home and caught up on notes before you arrived here. I will give you a moment to do so. We called this picture a mosaic. And we said a mosaic is made of a lot of different individual pieces. And what that means is the pieces all have to be different, so you end up with a picture that performs its function. It serves its purpose. You'll see that there are two kinds of proteins here. There are peripheral membrane proteins and integral membrane proteins, and these have different names. Integral membrane proteins are also called transmembrane proteins. Transmembrane proteins, and transmembrane means goes all the way across the membrane. Just like a transcontinental flight means you're going from New York to LA, all the way across the continent. A transmembrane protein is going to go all the way across the membrane. So that's another name for those integral membrane proteins. And they embed within that bilayer very specifically. There are polar parts and nonpolar parts of this particular protein. The polar parts line up with the glycerol, and the nonpolar parts line up with the fatty acid tails. Every transmembrane protein that embeds will embed in that way, where there are nonpolar parts in the middle and polar parts on the outside. This is another example of a transmembrane protein here on the far left. Yep. Glycerol. Nonpolar parts with the fatty acids. This is another kind of transmembrane protein that we have another name for. We call it a carrier protein. A carrier protein. And we call it that because it has a hole in it and it allows things to pass from one side to another. Every large molecule that has to pass like that has its own specific carrier. So, for example, I told you charged things have to pass through the membrane. They have their own pump that allows them to pass through the membrane. Membrane proteins that occur on only one side are called peripheral. Have you heard the word peripheral before? In what context? It's your peripheral vision. Where is your peripheral vision? On the side. So if you hold your hands up, right where you can finally stop seeing, like right there on the edge, that's your peripheral vision. Peripheral means side. So those peripheral membrane proteins are attached on only one side of the membrane. They don't go across the whole thing. They are on one side. And the last thing that you'll notice has embedded in this membrane is that on the inside of the cell or the outside, the last thing we haven't talked about are zigzaggy green things. We haven't said what they do yet. Are they inside the cell or out? Extracellular means outside of the cell. Carbohydrates attached to the surface of the cell as a way of saying, hello, my name is. They identify the kind of cell to other cells they encounter. So it's an ID tag of sorts. So that's what carbohydrates are there for. They're very important for intercellular communication. That's your membrane. Why do we do that as quickly as possible? Because the membrane itself isn't as interesting as the idea of how the membrane functions to keep things alive. So I'm going to ask you some questions about what should hopefully make sense. Let's see. Oops. So diffusion, when things diffuse, we say they move down their concentration gradient. That's the term we use for it, down the concentration gradient. So typically what I do is simply draw a hill. The top of the hill is high concentration. The bottom of the hill is low concentration. And it makes sense to you that if you were to set a ball down at the top of the hill, what does it do? Rolls downhill. Did you have to do anything to it other than just like set it there? Off it goes. Inclined planes, right? That's diffusion. The idea that it's just going to happen, moves from high to low, until it reaches a spot where it's level or even, and then it stops. Yeah, I'll do it that way. Osmosis is just a funny name for the diffusion of water. So if water is the thing that's moving from high concentration to low concentration, we call it osmosis. Facilitated diffusion is, again, still from high concentration to low concentration across the membrane, but it can't fit between the phospholipids. This is when they're too big. They can't squeeze in those little spaces. So instead, they use a carrier protein to move from areas of high concentration to low concentration. So on that first slide where we looked at that transmembrane protein that had a hole in it, if they're too big to fit through the little space and they have to use a larger space, it's facilitated diffusion. You can think about that in the same terms of a window screen won't allow your dog outside, but your dog can use the dog door at the bottom of the screen to freely pass one way or another. It's just a larger hole in the membrane to allow something to pass through freely. The idea of the dog door is the dog can go in and out at will, right? Whenever they choose. Same here. Facilitated diffusion, the transmembrane protein, it's so it can pass back and forth freely. These kinds of membrane transport that require no energy, they go from high to low, are called passive transport. They require no energy input. So you guys are good with that? When we move down the concentration gradient, we go from higher concentration to lower concentration until they've reached the same concentration, so 6 and 6.